Andhra Pradesh Rice Production Technology. Module 5 Disease Management. Overview. This module deals about disease management, which includes diseases that attack rice crop at different stages of crop growth and are more prominent in Andhra Pradesh. Objectives: to know about the causal organisms of diseases, symptoms and control measures of various diseases. Lesson 1. Diseases in rice: blast, bacterial leaf blight, rice tongue disease and sheath rot. Blast: Pyricularia griseae, P. oryzae. Symptoms: disease can infect paddy at all growth stages and all aerial parts of plant: leaf, neck and node. Among the three, leaves and neck infections are more severe. Small specks originate on leaves, subsequently enlarge into spindle-shaped spots, 0.5 to 1.5 centimeters length, 0.3 to 0.5 centimeters width, with ashy center. Several spots coalesce to more bigger irregular patches. Leaf blast: severe cases of infection, entire crop give a blasted or burnt appearance. Severe cases of infection, entire crop give a blasted or burnt appearance, hence the name blast. Severe cases: lodging of crop after ear emergence. Neck blast: neck region of panicle develops a black color and shrivels completely, or partially grain set, inhibited, panicle breaks at the neck and hangs. Control measures: avoid excess nitrogen fertilizer application. Apply nitrogen in 3 split doses. Remove weed host from bunds. Use of tolerant varieties. Burning of straw and stubbles after harvest. Spray phosphorus, fluorescence talc formulation at 0.5% from 45 days after transplanting at 10-day intervals 3 times. Seed treatment at 2.0 gram per kg seed with Captan or carbendazim or thiram or tricyclazole. Spraying of tricyclazole at 1 gram per liter of water or edifenfos at 1 ml per liter of water or carbendazim at 1.0 gram per liter. 3 to 4 sprays each at nursery, tillering stage and panicle emergence stage may be required for complete control. 2. Bacterial leaf blight. Xanthomonas oryzae, P. oryzae. Symptoms: water, soaked to yellowish stripes on leaf blades or starting at leaf tips, then later increase in length and width with a wavy margin. Appearance of bacterial ooze that looks like a milky or opaque dewdrop on young lesions early in the morning. Lesions turn yellow to white as the disease advances. If the cut end of leaf is kept in water, it becomes turbid because of bacterial ooze. Management: secure disease-free seed. Grow nurseries preferably in isolated upland conditions. Avoid clipping of seedlings during transplanting. Balanced fertilization. Avoid excess nitrogen application. Skip nitrogen application at booting if disease is moderate. Drain the field except at flowering stage of the crop. Destruction of wild collateral hosts. Avoid flow of water from affected fields. Grow tolerant varieties IR20, TKM6. Spraying streptomycin sulfate and tetracycline combination 300 grams plus copper oxychloride 1.25 kg per hectare. Rice tongue disease, rice tongue virus, RTSV, RTBV. Symptoms: plants affected by tongue exhibit stunting and reduced tillering. The leaves become yellow or orange-yellow, may also have rust-colored spots. Discoloration begins from leaf tip and extends down to the blade or the lower leaf portion. Delayed flowering, panicles small and not completely exerted. Most panicles sterile or partially filled grains. Management. Set up light traps to monitoring and attract vectors. Destruction of weed hosts on bunds. Leaf yellowing can be minimized by spraying 2% urea mixed with mancozeb at 2.5 grams per liter. Instead of urea foliar fertilizer like multipotassium nitrate can be sprayed at 1%, which impart resistance also because of high potassium content. In epidemic areas, follow rotation with pulses or oil seeds. Green leaf hoppers as vectors are to be controlled effectively in time by spraying monocrotophos at 1.6 to 2.2 ml per liter or fenthion or phosphomidane at 1.0 ml per liter or neem by applying carbofuran 3G at 10 kg per acre. In nursery, when virus infection is low, apply carbofuran granules at 1 kg AI per hectare to control vector population. During pre-tillering to mid-tillering, when one affected hill per meter is observed, apply carbofuran granules at 1 kg AI per hectare or spray monocrotophos at 1.6 to 2.2 ml per liter to control insect vector. Sheath rot, Sarocladium oryzae. Symptoms: irregular spots or lesions with dark reddish-brown margins and grey center. Discoloration in the flag leaf sheath. Lesions enlarge and often coalesce and may cover the entire leaf sheath. Severe infection causes entire or parts of young panicles to remain within the sheath. Unmerged panicles rot and florets turn red-brown to dark brown. Whitish powdery growth inside the affected sheaths and young panicles. Infected panicles sterile, shriveled or with partially filled grain. Management. Apply Ipomia or Prosophis leaf powder extract 5% or NSK 5%. Spraying of mancozeb at 2.5 gram or carbendazim at 1.0 gram per liter or benomil 0.5 gram per liter of water at flowering stage. Destruction of the infected plant debris by burning. Lesson 2. Diseases in rice: brown spot, fall smut and grain discoloration. Brown spot, Helminthosporium oryzae. Symptoms: occur in nursery as well as main field. Causes blight of seedlings. Leaf spotting is very common. Isolated brown, round to oval. Resemble sesame seed. Spots measures 0.5 to 2.0 mm in breadth. Coalesce to form large patches. Seed also infected. Black or brown spots on glume spots are covered by olivaceous velvety growth. Infection also occurs on panicle neck with brown color appearance. 50% yield reduction in severe cases. Management. Use disease-free seeds. Since seed soak or seed treatment with Captan or Thiram at 2.0 gram per kg of seed. Spray mancozeb 2.0 gram per liter or Edifenforce 1 ml per liter 2 to 3 times at 10 to 15 day intervals. Spray preferably during early hours or afternoon at flowering and post-flowering stages. Fall smut, Ustilaginoidea virens. Symptoms: only few grains in a panicle are usually infected and the rest are normal. Individual rice grain transformed into a mass of yellow fruiting bodies. Growth of velvety spores that enclose floral parts. Immature spores slightly flattened, smooth, yellow and covered by a membrane. Growth of spores result to broken membrane. Mature spores orange and turn yellowish green or greenish black. Management. Use disease-free seeds. Destruction of straw and stubble. At tillering and pre-flowering stages, spray hexaconazole at 1 ml per liter or chlorthalonil 2 grams per liter. 3. Grain discoloration. fungal complex. Symptoms: Drosclera oryzae, Curvularia lunata, Sarocladium oryzae, Phoma species, Microdochium species, Negrospora species, and Fusarium species. Grains are infected either after milk stage or after harvest or during storage. Infection may be internal or external, causing discoloration of the glumes or kernels. Dark brown or black spots appear on grains. Under humid condition, prominent fungal growth. Management: spray the crop with carbendazim plus mancozeb 1:1 at 2 grams per litre at boot leaf stage and 50% flowering. Store grains at 13 or 14% moisture.